let's talk about the gag protein. I explained all the conventions in the part on the Pol protein, so go watch that first, watch this second, even though it comes before Pol in the genome. All right. So, shown above, this is kind of a partly imagined, mostly real rendering of what the gag looks like before it is cut. So this is gag before the protease gets a crack at it. And so here are the parts in order. You got your matrix up here, which you can see is mostly alpha helical. You've got your capsid, which has two domains, an N-terminal domain here, which is mostly helical, a C-terminal domain here, which is mostly helical. Then you've got a helix to connect them to the rest. You see the pattern? And then you've got the nucleic capsid. Now, the nucleic capsid is going to carry a positive charge because it's got two zinc fingers. So it's going to hold on to two Zn2 plus little molecules. Yeah, zinc cations. Then you got your P6, which is this little thing at the end, and there's itty bitty spacers in between some of these things. Don't worry about the spacers. So let's take a tour of this molecule. We'll start with the matrix. So this is the matrix, and you can see right over here. There we go. So what are the important features of the matrix protein? Well, it has to bind to the membrane, right? It has to interact pretty directly with the membrane. So it's got some hydrophobic patches here, and those are going to help. But the main thing that's going to help it interact with the membrane is going to be the myristylation. And the myristylation is on the glycine at position 2. See the glycine at position 2? See how conserved that is? You would see a change in color if it was not a glycine there, but they're all nice orange, G. And so, yeah, that is going to be conserved. And so this myristylation is going to be the way that, in general, matrix proteins attach. This gag alignment over here is only HIV-1, because when you try, and like the chimpanzee versions, because when you try to align different retrovirus gags, basically this homology falls apart. They all fold up and make the same shape in the end, and they do the same job, but they have evolved so far and so fast and tried so many different things that you can't really get a sense of what's going on from an alignment if we go much broader than this. So there you go. All right, so we got the G2, that's the position of the polymethylation, and here is our matrix domain. And you can see the matrix domain goes up to right about here. And then you're thinking, there's a cleavage site, right? There's a cleavage site after the matrix. I'm like, yeah, of course there is. And the cleavage site is going to be a Q, which is a glutamine, followed by a small amino acid. Now, small ones are going to include things like A, sometimes G, sometimes T. So you'll have like a little QA, QV, QT, QG, something like that. And that will be the cleavage site. So there's your cleavage site. You can see the cleavage site lines up because it always does get cleaved. And this is a very good fit into the HIV protease, which allows this thing to be processed and to mature once it's left the cell. All right, next section. Let's look at the capsid. Now the capsid is the second part of our alignment up here, and so we're starting with the N-terminal domain. You see the N-terminal domain has a lot of N's in it, asparagine's, which, that's just a coincidence. The capsid is going to be really good at sticking to other capsids. Now, it's not the N-terminal domain that does that. It's the C-terminal domain. And so let's see where we get to the C-terminal domain. So the C-terminal domain is going to start right here. And so this part with these conserved basic and acidic amino acids, those are going to pair up. Also possibly these hydrophobic patches. I don't remember where the exact contact faces are, but you got a number of ways that these things can snap together like little Lego bricks. And so that's the C-terminal domain of the matrix. And you can see in HIV-1 it's pretty well conserved, and that part's actually decently conserved in some other ones because that's doing a job. Then we get to the end of the capsid, Sorry, it was conserved in the capsid. Yeah, I said matrix. Oh my goodness. At the end of the capsid, we expect to find another cleavage site. Do you remember the cleavage site? Yeah, you remember. Q and then a small thing. So like QA, QV, QM. That's weird. That's a little bigger than I was expecting, but looks like it works. And there's a QT there. Yeah. Little QI. QI is a little big. So we're just talking about something fairly hydrophobic in the second spot. And this is going to be tuned for each HIV protease is going to be a little better or worse. These things are also tuned probably to be suboptimal so that they don't get cleaved too fast. The cleavage needs to take a little while so that the virion is going to retain its sort of rigid structure. And then once it's far enough away from the cell, or it's been untethered or whatever, then it needs to make sure it's all cleaved. So yeah, the virus is slowing itself down potentially. Okay, after the capsid, the two domains, then we have our nucleocapsid. Now, you remember the nucleocapsid has two zinc fingers in there. How do you recognize a zinc finger? You probably remember from the Paul video. And it's going to be 4 cysteines or histidines in some combination, usually more C than H. Let's find them. And they should be invariant because you need 4 to make a little tetrahedral grip on a divalent cation. And so let's look through here. I see nothing, nothing, nothing. Oh, there's this salmon color for the C. And the H is going to be kind of a bluey, tealy color. So C, C, H, C. That's 1 zinc finger. And then C, C, H, C. That's 2 zinc fingers. And I don't remember if these things fold up like each zinc finger folds up separately, or if some of the C's and H's are participating, like from different ends in the same zinc finger. Three-dimensional folding is not something you would predict based on just looking at an alignment, but the alignment will show you what's conserved and what's important. All right, so we found our nucleocapsid, and we found the secret to why it's positively charged. And you can see that there's some stuff that changes in here, and there are some areas where there are little gaps that had to be put in because some of these sequences have extra amino acids at these positions, and that's fine. Let's see. So moving past it, we would expect to find somewhere near the end of the nucleocapsid a cleavage site, and you've got a potential cleavage site right there. You got another potential cleavage site right there. It looks like both of them are pretty conserved, your QA and your QS or QN. All right, and then you're going to have your P6 region at the end. Now the P6 region for HIV-1 is going to contain a thing called a late domain, which you either know about already or are about to learn about when we get to assembly. They come in several varieties. One is PP something and a Y. One of them is Y something something and an L, or a hydrophobic amino acid. And another one is either PTAP or PSAP, PTAP or PSAP. And these are going to bind to different components of the escort pathway. And it's all covered in the lectures. You can go look that up. But you can see them in the alignment. That's the cool thing. So look, here's the P, S or T, A, and then the other P is over here. That should probably be aligned over here with this P. But, alignment programs don't know what they're doing. They're just following their little algorithms to their heart's content. Yeah, as are we all, I guess, in some way. So there, that's your late domain right there, your little PTAP or PSAP. And then you just got a little sort of tail on here, and yeah, there you go. That's the story of the gag region. Now you've seen it in 3-dimensional structure and in amino acid alignment, and just for good measure, let's look at it rotating. So here, let me spread this out a little bit. So this is the capsid region only. And so you can see the capsid likes to first dimerize. So you'll have 2 capsids coming together. And then the dimers are going to make a hexamer. So you'll have a total of 12 little capsid proteins in here. The N-terminal domains are the parts that are sort of sticking up into the sky. If we rotate it, the C-terminal domains are the ones that are making a nice seal. So that, I believe, is the part that's going to form the outside of the capsid. It's just like a nice, hard, smooth surface that's going to be pretty hard for the cell to penetrate. Unless it does some terrible stuff like attaches ubiquitins and gets degrading proteases involved. I don't know. Yeah, we'll have to see on that. But there you go. So C-terminal domain is up, N-terminal domain is down, and that's the capsid protein. All right. Hope that gave you a different perspective on GAG. Thanks.